Okay, let's get started. Couple of announcements beforehand. First one is that there'll be some extra time to study on Thursday because there's not going to be a biology seminar. So noon to 1, you can spend all of your time studying for the exam. Some of you may have seen that there's another snowpocalypse apparently potentially coming at the end of the week. So if that is the case, which I doubt, if school is closed, the exam will be on Monday. If, however, it opens at 10, as this university has a nasty tendency to do, make sure you give yourself plenty of time to get here, because the exam will happen. No, there's not going to be an online test. Sorry. So any other questions about that? As usual, stuff that we cover today, so ask lots of questions and slow me down, is going to be the stuff that ends up on the midterm. Stuff we don't get to today will not be on the midterm. And again, it'll be a review on Wednesday. So any questions about what's happening? What's going to be on the exam? Yes, everything since the last midterm. Is it written yet? No. Other questions? Yes?
U3 is a helicase for which polymer?
Okay, so the question has to do here with U3, and I think, correct me if I'm wrong, you mean the U3 snRNP? Yes. So the U3 snRNP is involved in ribosomal RNA processing. And so it's actually lots of different things, but the main thing it does is trim the 5' end of your pre-ribosomal RNA. So it actually has nothing to do with splicing. There's U1, U2, U4, 5, and 6. They skip U3. 1, 2, 4, 5, and 6 are involved in splicing. 3 is involved in ribosomal RNA processing. Other questions? Again, feel free to email me, and I'll post on the discussion group as well. Yeah? So the question has to do with inosine being made from guanine or adenine. I actually posted something about that on D2L, so you can take a look there. Okay, so today we're going to continue talking about translation. Oh, sorry, yeah.
The U2AF is what's needed to bring U2 to the RNA structure, right?
So the U2AF, the U2 activating factor, is helping to bring U2 to your intron so that the branch point and all of the recombination can take place at that point to get splicing to take place. Okay, so, and by the way, thanks for being on your best behavior on Friday. The dean really liked being here and thought you were all really interactive. Cool. Thank you. So she said a really nice tweet, too, but I'm sure all of you are following her on Twitter. So, but yeah, continuing on, basically where we left off last time. Last time we talked about the activity of the ribosome, the transpeptidation reaction, etc. Today we're going to spend most of our time talking about how to start you actually getting your translation to take place. But just really quickly, sort of as review, we've got our large subunit of the ribosome, the small subunit of the ribosome, each of 3 tRNAs, which are bound to, in fact, right between the 2 large and small subunits. So this is those 2 together. The light green is the large. The dark green is the small subunit. That's looking at it from the side. Here, looking at it from the top. These guys are really squeezed between those 2 particular subunits. And then the messenger RNA is down at the bottom. And we'll talk a lot more about these individual binding sites here. They're the E site, the P site, and that A site, which is hidden by our nice little eye clicker thing here. So aminoacyl site, not surprisingly, that's where the tRNA binds that has the aminoacylated tRNA on it. That's the amino acid which you're going to add. The peptidyl transferase site, that's where you hook up your amino acid chain to the tRNA which is already there, and then the tRNA that used to have your peptide attached to it is in the exit site, and that then falls off. And so that's your elongating ribosome. How do you get to that point? You need to initiate. And this gets back to what we talked about last time in terms of all of the open reading frames. You've got to start at the right position and then move 3 nucleotides at a time in terms of reading your genetic code. So where you start is absolutely critical in terms of where you end up with. So this is a very general look at how translational initiation starts. We'll talk about the differences between bacteria and eukarya as we move on from here. And the obvious question is, Dr. Stedman, what about archaea? The answer is we don't know very much about archaea. Really fascinating question, and again, great stuff to look into, anybody looking for a really cool PhD project. Translational initiation is really all about getting the initiator tRNA. And last time somebody asked a question about this in terms of what happens if you have an AUG in the middle of your messenger RNA versus the AUG at the beginning. And I mentioned there's a specific tRNA which is the initiator tRNA. So it's actually a different tRNA relative to the normal methionine tRNA which you use to do extension in the middle of your messenger RNA. So this initiator tRNA has to find the appropriate AUG site, and then the large and small subunits of the ribosome have to come together. And this kind of makes sense if you just think about the ribosome structure that we had on that last slide. Those tRNAs, and particularly the tRNA which is in the P site, is really scrunched down in the middle of the ribosome. So it's really hard to get there. So getting this assembly of the initiator tRNA bound to the start codon and getting the 2 ribosomal subunits together, that's what you have to have in order to really start your translation process. So getting the tRNA, initiator tRNA to your AUG, getting the small and the large subunits to associate with each other. Now this is where the differences come in between bacteria and eukaryotic initiation. Bacteria are really quite straightforward. This is wonderful because, as we all remember, most of the ribosome is made up of what? RNA. A few extra proteins hanging out there, but it's almost all about the RNA. What does RNA love to do? Just like DNA does, base pair. So in the case of bacterial, but not in eukaryotic translation, you have a particular piece of your small subunit RNA right here, 16S, that's the small size, small RNA subunit, which has a specific sequence, 5' to 3', varies from bacterium to bacterium. And in this sequence, you have a completely conserved sequence, which is then complementary to what's called the Shine-Dalgarno sequence, also known as the ribosome binding site. So what happens, you get base pairing that happens between the small subunit of the RNA and the sequence which is complementary to that, this Shine-Dalgarno sequence. That brings the small subunit of the ribosome to a position right next to the AUG. And here it's shown as 8 nucleotides. It varies, sort of 8 to 10 nucleotides relative to AUG. So that gets your small subunit of the ribosome bound to the right position on your messenger RNA. And one of the things about having this very specific sequence that binds to the ribosome means you can have multiple places for a ribosome to bind in your messenger RNA. And so we talked about this when we talked about messenger RNA processing before. Bacterial messenger RNAs, unlike eukaryotic messenger RNAs, don't have caps. They've just got nice triphosphate here and are often polycistronic. So they can code for multiple completely different proteins. And the way that they do that is they've got these ribosome binding sites or Shine-Dalgarno sequences just 5' to where your AUG is where you can actually start. We mentioned this before, but it bears repeating. When you look at bacterial protein production, going all the way from your DNA to protein, you have transcription, where you take DNA and make it into RNA. But instead of all this wild processing stuff that we talked about, different U's, splicing, capping, tailing, etc., what you have in bacteria is as soon as that messenger RNA is made by the RNA polymerase, a ribosome will bind to that with its ribosome binding site, and you start translating. And so you have transcription and translation happening more or less simultaneously, or at least one immediately after the other. You can't have it simultaneous because, of course, you have to transcribe before you can translate. But transcription and translation are very closely coupled in this case. So how do we get from this binding, again with the Shine-Dalgarno sequence, binding to the small subunit of the ribosome, to getting that initiator tRNA bound to the right position? Now this is, well, overheard people talking about studying already. Let's have a little bit more alphabet soup. So these are the IFs. So we talked about the TFs already, right? Our transcription factors. Now we're going to talk about IFs. So the IFs are the translational initiation factors. Fortunately, there are only 3 of them, unlike only 3 for bacteria. Way more in eukaryotes, but we're not going to talk about most of them. Because these are, again, not terribly surprisingly, 3 of the initiation factors interact with the 3 different tRNA binding sites in the ribosome. So IF1 binds to the A site in the ribosome and basically makes sure that the initiator tRNA can bind to the T site, because all the RNAs, when they come into the ribosome, are going to want to bind to the A site, because that's where you want to have elongation taking place. Every time you get a new tRNA added to the ribosome, it's going to want to come into the A site. So the IF1 is really important for blocking the initiator tRNA from binding to the wrong part of the ribosome. And then we'll skip IF2 for the time being, probably the most important one. And IF3 basically says, hey, we're not ready for the large subunit of the ribosome to bind here yet. So 1 and 3 are basically saying, hey, hold off. We're not ready to go yet until we have our initiator tRNA, which comes in with IF2. And IF2 is really the critical one here. IF2 binds to, not surprisingly, the initiator tRNA, and is also a GTPase. And so we talked about GTPases when we talked about signaling before. GTPases bind to GTP, and then when they have GTP hydrolysis, there's a change in structure. And in this particular case, and as we'll see for a bunch of different times when we talk about translation, this change in the structure with GTP hydrolysis is going to release that initiator tRNA at the right position in the ribosome. So once you have that tRNA, which is in the right position, again, your initiator tRNA, then IF1, IF2 come off, and IF3 come off, and then you can have the large subunit of the ribosome, and then you're ready to translate. So that's translational initiation, in bacteria. Yeah?
Just to clarify, IF3 and IF1 are bound to the subunit until IF2 comes in, and then when it does, when it binds to a initiator tRNA, GTPase initiates translation, and then IF1 and 2, or IF1 and 3 get kicked off.
Okay, so that's very close. So pardon me for not repeating the question, but the only thing that I would differ with you on there is that the initiation of translation I would think of as being the first peptide bond which gets formed, and that can only happen after you have the large subunit of the ribosome coming in. And so it's the GTP hydrolysis that happens by IF2 which will release the initiator tRNA in the right position and basically, of course, this is an oversimplification, kick off IF1 and IF3. Basically, all of the initiation factors are going to come off after you have that GTP hydrolysis. But GTP hydrolysis only happens if you have the appropriate base pairing happening between that initiator tRNA and the start codon. Okay, other questions on this initiation? Yes?
So then after that sequence, the large subunit can come
Exactly. So after you have all the release of those initiation factors, the large subunit can come in, and very importantly, IF1 is gone, so now you can get your next amino acid with that next tRNA bound to the A site. Yeah?
What has to happen for the large subunit to bind?
What has to happen for the large subunit to bind? IF3 has to be gone. More questions? No? What does he mean when he asks about more questions? So we're still trying for 100%, right? That's our plan. No. How many different binding sites are there for tRNAs in the ribosome? 1, 2, 3, 4, 5. He loves to have these kinds of questions because it's so easy to write answers for. Don't trust him. Don't trust him. Never trust him. Ten, 5, 2, 1. I swear you guys are messing with me. This is okay. What are the 3 sites? A, P, E. Yeah?
There's confusion on this image, though.
Pardon?
Because the binding site really, only shows 2. Like, it says mRNA binding sites.
Well, so this is about the tRNAs,
Right.
so not mRNA. If I'd asked about RNA binding sites, then we'd have 4, because you'd have the mRNA as well. Yes?
Does the E site actually bind before exiting?
The E site is a binding site for a tRNA, yes. Okay, so we're at 81%. I think we did better last time. Okay, so what about eukaryotic transcriptional initiation? Sorry, translational initiation. I will make that mistake probably far too often. Here, we don't have a nice Shine-Dalgarno sequence. We have an AUG, which is used, and then a few nucleotides which are quite well conserved next to that AUG, where you actually start. And the reason that we know this is because Marilyn Kozak and some of her colleagues basically looked at a whole bunch of RNA sequences and then just compared them all to each other to find out where the start for translation was. And what they found was, not surprisingly, 100% of the time you had an AAU and a G here. And then quite often you had a G here, sometimes you had a A or G here, and sometimes you had a C here. And this is probably because many of your eukaryotic RNAs are really quite long, and we're also going to talk a lot about the excuse me, process of translational regulation in what's called the untranslated region. So it's between the cap and where you actually start. And so what you have to have, as we'll see, is the ribosome associates with the cap, and then it's got to move along the RNA to get to the right place to start. So the bacteria, again, got it way easier because they can just have this Shine-Dalgarno sequence that you can have the ribosome bind to. Here, it's a much more complicated process. And this is the ridiculously complicated process that I'm not going to talk about because we're going to oversimplify it and move on here. Just a quick thing here, and my apologies, this didn't end up in the PowerPoint that I tried to post last night. Not quite sure how that didn't happen. But here, there's also a lot of cancer inputs in terms of the regulation of translational initiation. Yes?
So the Shine-Dalgarno sequence is upstream from the bacterial initiation site.
So the Shine-Dalgarno sequence is going to be upstream, and that's 5' on your messenger RNA from the AUG where you're going to start. And one thing which is actually quite good about that, thank you for reminding me, is that because you've got that very specific sequence, the Shine-Dalgarno sequence is going to be base pairing with your ribosome. That's why bacteria can often use slightly different start codons, unlike what you have in eukaryotes. Because if you've got that Shine-Dalgarno sequence, right next to it, as long as it's something that's close to an AUG, so you can have GUG, you can have CUG, you can still have your initiator tRNA that's going to bind there and start there. Whereas if you're scanning along the RNA, looking for your AUG, like we have here in the eukaryotic system, that's going to be a lot less likely, because it's got to scan until it gets to that very specific position. And it's really important that you find that AUG there. Whereas the Shine-Dalgarno sequence, you've already held the ribosome right there. So here, it's a little bit different. You actually start out here in eukaryotic systems with a small subunit of the ribosome already bound to the homolog of initiation factor 2. Now here, little e in any of the systems just stands for eukaryote. So eukaryotic initiation factor 2. So little e IF2, little e IF1, little e IF3, etc. And conveniently, EIF1, there are a bunch of different ones, EIF1A, has a very similar activity to IF1. What's IF1's activity? Blocking the A site. IF3's activity? Blocking the large subunit of the ribosome. So EIF3, EIF1 are serving exactly the same roles. And conveniently, EIF2 also serves a very similar role, except it's got a whole bunch of extra proteins, IF5B, et cetera, et cetera. That's all those extra blobs that you saw in the last image there. So this process, again, 1, 3, EIF2, very, very similar, still binding to your initiator tRNA, waiting until it finds the right place. But we're not coupled transcription translation here. This is a modified, capped, tailed, spliced messenger RNA that's got exported to the cytoplasm. So somehow now you have to get this small subunit of the ribosome and, very importantly, your initiator tRNA to the right place. How does that happen? You have initiation factors that are bound to your cap structure. So particularly here, these are the important ones. And again, this is very different between bacteria and eukarya because bacteria don't have these caps. So cap proteins that we're going to care about are EIF4E, which is literally binding to the cap structure, and EIF4G. Now, what's unfortunately really ridiculously confusing about this is that EIF4E and EIF4G are also known as EIF4F. So which ones am I going to use for the exam? I don't know. It's not written yet. So I will try and be specific about these things. But I just wanted to point this out. This is again, it's a ridiculously confusing nomenclature, and no, I didn't make it up. So EF4E is your cap binding. EF4G serves as a bridge between your poly-A tail, and the way it serves as a bridge is a poly-A tail binding protein that it binds to, as well as EIF4E. And so what this means is your messenger RNA is actually very much a looped structure. And we'll presumably get back to looking at that at the end or middle of lecture today. Looking at that overall looped structure between the 5' end and the 3' end of your messenger RNA, which are very close to each other. So EIF4G and EIF4E are all together with this messenger RNA that's been exported appropriately to the cytoplasm. So it's the small subunit of the ribosome which is now going to associate with this EIF4E and EIF4G, but that's at the 5' end. That's where your cap is. Your AUG is over here. So somehow you have to get your small subunit of the ribosome from the 5' end of your messenger RNA through this whole as I mentioned before, untranslated region, also known as UTR, until you get to the right place. In many messenger RNAs and in many of these UTRs, there are a whole bunch of secondary structures. So they're stem loop structures, they're pieces where the RNA is bound to itself, etc. And in order to get the small subunit of the ribosome, which has this messenger RNA binding piece in it, through that small subunit of the ribosome and get you to the right place, you need a lot of RNA helicase activity. So that should sound really familiar. Where else do we see a lot of RNA helicase activity? In the spliceosome, in terms of all the rearrangements of your small nuclear SN RNAs. So lots of RNA helicase activity in order to get the small subunit of the ribosome and the initiator tRNA to the right place. Once you're here, it's great. Codon-anticodon binding, EIF2 hydrolyzes your GTP. That falls off. Once you have that hydrolysis, same thing happens as what you had happen in the bacterial system. You have EIF1A that comes off, EIF3 that comes off, large subunit of the ribosome comes together, and now you're ready to go. You're ready to elongate. So elongation step, again, just as we talked about before, we've got our aminoacyl tRNA comes in, binds in the A site. After you have the binding in the A site, then you have peptide bond formation that happens here. The amino acids that are hooked up to the tRNA, which is in the P site, now have a peptide bond to this amino acid, which is present in the A site. This is the peptidyl transferase reaction, all catalyzed by the large subunit of the ribosome, and apparently all just by the RNA. No proteins involved there. And then the ribosome needs to move, because now we've got our growing polypeptide chain here in the A site. There's nowhere to get that new tRNA bound. So there's actually a two-step process. First, the large subunit of the ribosome moves, then the small subunit of the ribosome moves, and once that has happened, you now have an open A site. This tRNA that used to be in the A site is now in the P site. Now we can have a new tRNA binding here, and this process continues. So it just chugs its way along, and eventually making your full RNA chain. This can happen with just the ribosome. It's incredibly inefficient without the ribosome. So there are a couple of other things that you need to make sure that this happens with a rapidity that actually makes sense for the cell. A couple of things have to happen here. Again, more alphabet soup. But now instead of the IFs, we're EFs. Okay, what's the heck's the E? Okay, initiation, elongation, termination, and we'll also talk about release factors. These are now your translational elongation factors. Elongation factor G will bind to the large subunit of the ribosome once it's made this little translocation step, and through GTP hydrolysis, basically pull the small subunit along. So EFG in bacteria and eukaryotic elongation factor 2 are the ones which help move the ribosome along, moving the ribosome along your messenger RNA. Again, you don't absolutely need them. You get this activity in the absence of these factors, but they speed things up considerably. And again, it's all about GTP hydrolysis causing a conformational change, which will then be coupled to another conformational change. Do you have another question in the back?
Yeah, you said that EFG2 moves the lower subunit, the smaller subunit, and that EF2 moves the larger unit?
Okay, so here, this is, anytime you got a little E, what does that mean? Eukaryote. So this is the bacterial case, is EFG, and the eukaryotic elongation factor 2. They're both doing the same job. EFG is in bacteria and elongation factor 2 in eukaryotes. So you could just change this label right here, EFG to EEF2 for a eukaryotic system. So they're doing the same thing, which is helping move the ribosome along. Mostly seems to be involved in moving the small subunit along the messenger RNA, but we can just think of that in terms of a translocation, helping to translocate, move the ribosome along relative to where your tRNAs are and where the messenger RNA is. Yeah?
So you said this already, but is the small subunit moving first or the large subunit?
So in terms of which one moves first, it seems to be the large subunit moves first, and then the small subunit kind of catches up with it. Okay, so that's elongation factors. In terms of, you may be wondering why is it EF2, right? Go figure. There's an EF1 as well. So what's EF1 doing? I think it's easier to think about EF1 first, and then we'll get back to EF2. It's just the bacterial version of this. This is basically doing exactly the same job as what we had with initiation factor 2 or eukaryotic initiation factor 2. What it does is it associates with your aminoacyl tRNA and makes sure that it has the right base pairing interaction here. If it does have the right base pairing interaction, then you have GTP hydrolysis. This guy continues. You keep moving along. If, on the other hand, it's not the right base pairing interaction, you don't have GTP hydrolysis, and it falls back off. So it's this sort of quality control, making sure that your base pair interactions between your codon and anticodon are actually exactly correct with, of course, all the wobbles that we talked about last time. And so it's that structural interaction there. And again, making sure that it's the right interaction, the appropriate base pairing interaction with your codon and your anticodon. And it turns out we know a lot about the structure of this, and we'll take a look at exactly what's known about the structure of this in just a second. Yeah?
So you can have translation without these, they increase the efficiency or fidelity?
So the question is, you can have translation without this. They increase your rate and fidelity. Yes, and so the rate is from your EFG, elongation factor 2, and the fidelity is from EF1 in eukaryotes and EF2 in bacteria.
So EF1 would be the proofreading mechanism.
EF1 is helping to make sure you've got those appropriate interactions. Yeah.
What drives the tRNA landing in the right site? So assuming that you're just in the cytosol and you have billions of tRNAs floating around, is it like osmotic pressure? What drives the individual tRNAs to the A site?
So what drives the question here is basically what gets those tRNAs into the A site. The answer is it just seems to be diffusion, which if you think about it, that's going to be really inefficient. So basically you got to go through all 20 to make sure that you've got the right one. And one of the things that I'm going to mention later or can mention now as well, translation is actually one of the slowest processes that we've talked about so far. Replication, really quick. Transcription, pretty quick. Translation, ridiculously slow. And that's probably because of exactly that reason, that you've got 20 different amino acids which could come in and be associating with that particular position. So it does just seem to be diffusion. And it means you also have to have a lot of these RNAs which are out there. And we'll talk more about this a little bit later on. But if you've got a whole stretch of exactly the same amino acids, one right after each other, that slows down your translation considerably, because it's just it's a statistical thing. How often you can have exactly that same amino acid which is going to be available. So yeah, this is the really slow step of translation. Yeah.
Does that mean EF2 is associated with all the different ones then, or does it only associate right at the corpus site?
Okay, so the question is, and well noticed, is whether EF2 or elongation factor 1 is associated with all of the tRNAs or just right as it comes into the ribosome. It's associated with all of them. So all of those tRNAs, they have And when we look at our animation a little bit later on, it's not attached to it. But they all are going to have those elongation factors associated with them. OK. More questions? What does he say when you have more questions? Quick, quick, quick. Hand up, hand up. OK. Let's do another quicker question. What seems to be the main role for ATP hydrolysis in translational initiation by eukaryotic ribosomes? Ribosome translation from codon to codon, removal of secondary structure in messenger RNA, checking to make sure codon-anticodon pairing is correct, coupling to transpeptidation, association of the large and small subunits of the ribosome. Oh, it's great. I have up here, I've got 5 different options. First votes were 5 different votes. Still aiming for 100% here. Ten, 5, OK, not too many people like B or D. Actually, the least people like B, which is unfortunate because that's the right answer. So the key here is ATP hydrolysis. What are all those other proteins associated with? GTP. So what's the ATP hydrolysis for? Whoever answered this correctly, well, 4 of you answered it correctly. It's the RNA helicase activity. RNA helicase activity, getting the ribosome to the right place. Also about translational initiation here. So the answer is B. It's removal of secondary structure in your messenger RNA. So that's the correct answer here. No, I'm not trying to get zeros for anybody here. Obviously, I didn't do a good job of explaining it. So
removal of secondary structure
in the messenger RNA. So ribosome translation, translocation is from codon to codon is what? So it's going to be GTP, and what's the protein that's involved in that? Elongation factor 2 or G, right? So checking to make sure base pairing is correct. Which one's that? Elongation factor 1 or EFTU. Coupling to transpeptidation. The ribosome does all that. We don't need to bother about it. And associated to the large and the small subunit. Is there any hydrolysis that takes place there? Well, to some extent, yes. It's the GTP hydrolysis by what? Initiation factor 2. Because that's the hydrolysis which then allows EF3 to come off, or IF3, excuse me, to come off. See, I get confused about all these things as well. So GTP hydrolysis by initiation factor 2, or eukaryotic initiation factor 2. Because that's when you've got your initiator tRNA, which is there. So now I wanted to show you this, I think, really amazing movie, which some of you have seen before here. So this is now looking at that elongation process where you have EFTU coming into the ribosome in the A site, bound to one of these aminoacyl tRNAs, and the whole structural process which happens there. So here's our EFTU coming in, with your tRNA coming into the ribosome. This is all from structural work. So here's your protein, and there's the tRNA over here. So now we're checking to see if the base pairing interaction is proper here. I'll get to it right at the end. So if we have appropriate base pairing interaction, then GTP hydrolysis is going to happen in this EFTU here. Here's that amino acid, which is the end here. We have interactions between the small subunit of the ribosome and EFTU. That's the amino acid which is present at the end here. Lots of structural changes which are going on here. Most importantly here is now we're going to have our GTP hydrolysis. We've got our right interaction because of the binding here. Now we can have GTP hydrolysis that takes place. There's a little phosphate running away here. After our GTP hydrolysis, we have a structural change in EFTU, which is now going to fall off. Now this tRNA is released, moves the piece over to here. We can now have transpeptidation taking place, and your exit tRNA is removed. So, and that is more or less state of the art what we know about the ribosome in terms of the EFTU. So there's interactions here with the small subunit, between the small subunit and the tRNA in the A site, as well as the base pairing interactions which are happening down here. Yeah, your question. Where is the growing peptide chain here? It's going to be right here. So attached here, and then there's a little hole in the ribosome which goes up here, which you can't see in this particular image. Green here is in the P site. It's in the peptidyl transferase site. It was the yellow one that just left. That was the one that was in the E site. And after this, you have translocation. There's another video I could show of that, but we're not going to go there. And then we have this guy moving over into the P site, this into the E site, et cetera. Yeah?
So the conformational change that released the tRNA that's in the A site, it looked like it was actually coupled to the E leaving. Does that conformational change push the E tRNA out?
So the question here basically has to do with, does the conformational change that happens when the A site tRNA moves in, is it also coupled to the E site tRNA leaving? It certainly looks as though it is in this animation. Whether it is or not, I'm absolutely not completely sure. I believe that it is, but I can check and see how much is known about that particular process. Okay, so if you'd like to look it up, this is where that particular paper was published. So now we've done elongation. What do we need to do next? Termination, exactly. So what are we going to have? More alphabet soup. So unfortunately, these are not called termination factors, which I would prefer that they were, but it would then be confusing with all of our transcription factors that we've talked about already. So they're called release factors as opposed to termination factors. So what happens when your ribosome gets to a termination codon? There's no tRNA that binds to this termination codon. So all 20 of those amino acids come in and check, and okay, nothing is working here. Then you have a release factor, which is going to bind to one of these termination codons. Turns out there are different termination factors, not all of them, say release factors that interact here. And then once you have this interaction, this apparently tricks the ribosome into trying to make a peptide bond to the non-existent amino acid, which is present at the end of the non-existent tRNA, which is in the A site. And what that does is then forms a peptide bond to water, and you have now a carboxy terminus and a released protein, which is present here. And once you have your amino acid chain no longer bound to your tRNA, this comes out of the ribosome very rapidly, gets folded up, becomes a protein, does its own thing. On the other hand, this last step where everything falls apart, this requires considerably more things going on. And let's take a closer look at that here. So release factors, here is one of those release factors here. Release factor 1. Spent a little while talking about what each of these does. But basically, this guy pretends to be a tRNA sitting down in this A site, but it doesn't have an activated amino acid at this end. And so here you get your transpeptidation to water, you release the peptide chain. But now, after this has happened, you have this kind of situation here. So released peptide with a release factor here, and then 2 tRNAs, one sitting in the P site and one sitting in the A site. Now you need to remove all of these things from the messenger RNA, all your tRNAs, which are bashed down in the middle here. How can you get that to take place? Well, it's our friend GTP, right? So GTP hydrolysis and GTP binding then take your release factor, unfortunately hidden over here, after you've had this transpeptidation to water, releasing your amino acid chain. Now we have GTP, and in this case it's GTP binding, which then allows this whole complex to come apart. And I'm not going to ask too many details about this process. The EFs, we've got enough alphabet soup to be worrying about here. But once we have the removal of this first release factor from this site, then we have that same translocation factor that we had before, also binding to GTP, which can push the E and P site RNAs out of the ribosome. And once these have been pushed off, now you can have initiation factor 3, which can come back down and bind to the small subunit of the ribosome. What does initiation factor 3 do? Blocks the large subunit, exactly. Blocks reassociation of that large subunit. And so this is the end of the cycle, now ready to start again. And this small subunit of the ribosome can go, and if it's a eukaryotic one, go and find the cap. If it's a bacterial one, Scheindel-Garnell sequence to bind to the next ribosome binding site. Yes?
So for our purposes, there's one release binding factor. The elongation factor makes the ribosome continue as if it normally would, and then initiation factor 3 binds dissociating the entire ribosome.
Yeah, so that's a nice, again, as always with these things, a ridiculous oversimplification, but good enough as far as we're concerned. So we have a release factor that comes in, pretends that it's a tRNA, causes the ribosome to terminate translation, which is just making a peptide bond to water. So after that happens, you need to dissociate this whole complex. That happens through EFG and GTP. And a bunch of other factors, but I'm not going to ask you to remember all the names of them. Yes, yay, happy, good. Okay, so one of the things as far as these release factors is concerned is, did they really look like tRNAs? And the answer is, yeah, they actually do kind of look like tRNAs. So here's a tRNA structure, and here's one of those release factors. It's got a pretty similar structure to it. This is where your activated amino acid would be. This is that part of your release factor, which is not an activated RNA, so we can have this. So now I want to look at yet another animation. This one is missing a lot of things, including EFTU, but gives you a bit better idea what's going on as far as this process. So here we have our messenger RNA, which is now going to get translated. It's just come out of the nucleus.
From the DNA out of the nucleus to a ribosome for production of the particular protein that this gene codes for.
I don't like the commentary here. So here's our cap structure. We're moving along the RNA. Here's all of our tRNAs, again, just floating around. And there's our extending polypeptide chain. As soon as it comes out of the ribosome, starts to fold up into secondary structures, and there we just found our termination codon, and here it is. So now we've got our small subunit of the ribosome binding to first the cap, translocating along, burning ATP in the process, and now chunking along your messenger RNA 3 nucleotides at a time. Here are tRNAs, which are missing what? Your EFTU or your elongation factor 2. So they should all be associated with those as well. And the little red dot is that activated RNA, which is at the end here. So here we've got always the right one coming in and associating. Of course, that's not always true. We've got lots of other ones which are coming in here. And this codon every 3 nucleotides right down here in the bottom. Here's that tRNA coming in, making the association, and extending each of these pieces. And again, this would be a lot slower because each of these is being tested here. We don't see the large subunit of the ribosome. It's been taken off in this particular image so you can actually see what's going on here. Again, here's those amino acids coming out of the ribosome. As they come out of the ribosome, getting folded up into this whole process. A site, E site, E site. Again, in that elongating process. Again, it'd be nice if we had more of the tRNAs bouncing on and off here, because hey, wow, amazing! Each and every single one is exactly the right one. And again, we're also missing the elongation factors in this process. Again, moving along, your messenger RNA, 3 nucleotides at a time. You also can see here that the two different size pieces, the large and subunit, are moving relative to each other. That should be shown here. Here it's just chunking along one at a time. This should be moving independently of each other. First the large, and then the small. This is the small one that we're looking at right here, the small subunit of the ribosome. And there's our peptide chain, which is being made and being folded here on the outside. So we can go back to presentation here. One of the things I'm not going to show you is the, sure, yeah.
So the tRNA binds to the mRNA via 3 base pairs, correct?
So yes, the tRNA is going, the codon and the anticodon are going to interact with each other with 3 base pairs, one of those which is a wobble. So sometimes it's going to be the exact base pairing, some quick base pairing, sometimes it won't be.
And already attached to the tRNA before it comes into the ribosomal subunits is the amino acid that's going to peptide bond to form the protein that's exiting the large subunit.
Yes, so the tRNA that comes in is an already charged tRNA, which has that amino acid attached with that high-energy ester bond that the aminoacyl tRNA synthetase that we talked about last time has put onto it. So those are all your charged tRNAs that are associated in that case. Okay, what we're not going to do is talk about this interview with Harry Noller and how he loves vintage race cars and fun stuff. He can probably buy a nice new one with the $3 million he got from Google. So working on it. Whether I can buy the sports car or not, I'll have to ask my wife. So as I mentioned when we first talked about initiation in eukaryotic messenger RNAs, these are all looped structures. So the 3' end and the 5' end are right next to each other because EIF4G has this bridging activity. It interacts with EIF4E, which is bound at the cap, and the poly A binding protein, which of course is bound at the 3' end of your messenger RNA. Now, why is this so useful? It's because when you have translation that takes place, your ribosome moves all the way around here. It gets to a stop codon. What happens? You have all the release factors, and the small subunit dissociates. It's right next to the cap, so it can reassociate and start going around again. And so one of the things that you see whenever you look at an actively translating messenger RNA, and this is the micrograph of one of those down here, they always have lots of ribosomes associated with them. And so this process, once you've started translating, this can go and go and go and really amplify the amount of protein that's being made. We talked about amplification before. It's that making of a RNA from a particular gene. You can make many copies of that RNA. Once you have that RNA, you can make many copies of the protein as well from that. And how that happens, it's the polysomes, so polyribosomes, and multiples of those making multiple copies of your protein. Yes?
Is this mRNA being recycled?
Okay, the question is the mRNA being recycled? I'm not sure I quite understand your question, but after the messenger RNA has been translated, then at some point it's going to get degraded into individual nucleotides, and at that point it's recycled. Yeah, they're cycling ribosomes going around. I was a little bit confused about that. So one of the things about translation is, A, it's really slow, again, because of all of these tRNAs which have to associate in the A site. The other thing is it's incredibly energy dependent. So you have massive amounts of mostly GTP hydrolysis, which is taking place in terms of elongation, EFTU, elongation factor 1, making sure that all those tRNAs are the appropriate ones, EFG, elongation factor 2, making sure that you're moving the ribosome along properly, the ATP that you need in terms of getting rid of all that secondary structure, but also getting back to your question about the charged tRNAs. How do you charge your tRNAs with the amino acids? There you also need nucleotide hydrolysis. So it's an incredibly energy-dependent process. Translation is probably the most energy-dependent process, at least of the molecular biology processes that we're going to talk about inside the cell. And so what that means is the cell wants to have ways of making sure that if something is wrong with your messenger RNA, that you don't burn all of these nucleotide triphosphates and waste all of this energy. So there are a number of processes for dealing with problems that happen with your messenger RNA. It's not really, I wouldn't call it really recycling, but if there's a problem with your messenger RNA, then you basically want to kick the recycling into high gear, or actually maybe more of a garbage disposal than a recycling process. So there are a couple of ways of dealing with this. One, of course, is that to protect your messenger RNA in eukaryotes, you have a nice circle, i.e. the ends are not available to be being degraded. Another thing that happens, we'll talk about this now in a little bit more detail, is what's called tmRNA. So we had all different kinds of RNA so far. T and M, it's basically a combination of transfer and messenger RNA. The gene name for it is ssRA, but I'll just talk about it in terms of tmRNA. So what does tmRNA do? Basically, tmRNA is a bacterial mechanism for dealing with broken messenger RNAs. And a broken messenger RNA is really problematic for a number of reasons. The first one, if you have a broken messenger RNA, you actually can't get a release factor to come into the A site because it's just gotten to the end here. It's stuck. And so if you need this ribosome to be making other proteins, it's a big problem. But also, again, this is clearly an incorrect RNA because it doesn't have a nice stop codon at the end. So if you have one of these ribosomes that has a broken messenger RNA in it, just has a 3' end here, then the tmRNA can associate with this. Now, tmRNA is a combination of a tRNA and a messenger RNA. The tRNA part has an alanine attached to it, and the mRNA part is just this tail that basically serves as a template for the ribosome to put in more amino acids.
Is it always an alanine?
It's always an alanine in this case. The other nucleotides that are along actually code for different amino acids. And that different amino acids here, there are 11 amino acids that are coded for here. Again, not all of them alanines, but basically this stretch of amino acids here, I like to call them the eat me signal. Because if you have a protein that has that stretch of amino acids at the end, it means it's come from a broken messenger RNA. So this protein is highly unlikely to be appropriately functional. So there are specific proteases that will come and bind to this part of your protein and chew up the whole protein in this process. So this is a way of dealing with broken messenger RNAs. Again, it's a bacterial specific mechanism. Another thing that I wanted to mention is we talked a little bit about the amino acids, how many amino acids we have in the normal genetic code, 20. How many different amino acids do we have in proteins? Actually about 24. So how the heck do you get these other amino acids? There are lots of different mechanisms for doing that. Probably the most important is something called selenocysteine. Selenocysteine has a selenium in it. That's why we actually need to have selenium in our diets. There are a couple of enzymes. I forget what they are. I'll try and post them on D2L. Where you have to have selenium in their active site, and if you don't have them, you're dead. So how do we get one of these unusual amino acids, and in this particular case, selenocysteine, which is added? Turns out that there's a specific selenocysteine transfer RNA that gets serine charged onto it by the serial aminoacyl tRNA synthetase, and that gets converted to selenocysteine by another enzyme, which will bind to this selenocystyl tRNA and change that to selenocysteine. Then there's a third protein which associates with this specific tRNA and brings it into the A site of the ribosome only if there's one of these extra structures which are here. All of you remember your genetic code, right? UGA down at the bottom here, what's this normally stand for? Stop codon. Here now, we have a selenocysteine tRNA that can bind here if it's brought in by the appropriate protein due to some kind of secondary structure in the RNA downstream of where this translation is going to be. So it's a normal tRNA synthetase, but a different tRNA and a different protein. Again, not terribly surprising. It's a GTP binding protein, hydrolyzes once we have all these appropriate things. Once selenocysteine is added here, that selenocysteine is just hanging off on the side chain. The amino group and the carboxy group of this particular amino acid are identical to each other. So it just gets included as normal parts of the protein. I'm going to skip program frame shifting because it's viral and totally cool, and we'll talk about it next term. Because I wanted to finish up talking about both nonsense and non-stop mediated decay. So we had some questions about that before. So a nonsense codon, what's a nonsense codon? It's a stop codon, but it's a stop codon that's in the wrong part of your messenger RNA. If you have a stop codon in the wrong part of your messenger RNA, your ribosome is going to stop before it gets to the end of the protein. If it stops before it gets to the end of the protein, and this is only true in eukaryotic systems because we've got a bunch of caps and splicing junctions here. If your exon junction complex, you remember the exon junction complex was one of the things that said, hey, this is a good messenger RNA to be exported. That messenger junction, the exon junction complex on the messenger RNA tells the ribosome, hey, you haven't gotten to the end yet here. And if you haven't gotten to the end yet here, that means there's a problem with this messenger RNA. If there's a problem with this messenger RNA, then we need to get rid of it. And the best way to get rid of a messenger RNA is to chew off the cap structure. And why is that so useful? First thing is the cap is there to protect your messenger RNA. Second thing, it's making that loop to the 3' end. So it's really doing a very good job of protecting it. And there are lots of exo and endonucleases that are present inside the cell. Last one I wanted to talk about is sort of the eukaryotic equivalent to tmRNA. If you don't have a stop codon, so the flip side, some of you lost your stop codon. You still have a poly A tail. If you have a poly A tail, what's going to happen? That's going to get translated into a whole bunch of lysines because the genetic code for lysine is AAA. Once you have this translation, gets all the way to the end of your messenger RNA, that then will associate with these extra release factors That whole lysine tail at the end says, hey, just like with the tRNA, this is the eat me sequence. Something's wrong with this RNA. And our exosome that we talked about last time in terms of recycling of all the introns will also now chew up this messenger RNA. Chewing up the 3' end is not as good as chewing up the 5' end in terms of dissociating everything, but still, once you have this end that's broken up, all the rest of your amino acid and your RNA is going to be degraded. So that's where we're going to stop today. That's the material which is going to be on the midterm. The rest of the stuff we'll talk about next week.